All right, welcome everybody. Thanks for taking the time to come down here, and thanks to the folks on Zoom. Before we get started, I'd like to just say that the Field Museum acknowledges that it resides within the traditional homelands of the Three Fires Confederacy, the Ojibwe, Odawa, and Potawatomi. The museum recognizes and is grateful for the original peoples who laid the foundation for the city of Chicago and for the diverse indigenous nations that reside in Chicago today. It is my great pleasure today to introduce Thais Vasconcelos, who's coming to us from not too far away over at the University of Michigan in Ann Arbor, where she started just last year as an assistant professor and curator of the herbarium and the EEB department there. Thais did her undergrad and master's at the Universidade de Brasília in Brazil, and her PhD work at the University College in London on floral evolution in the flowering plant family Myrtaceae, which you see here, and then did postdocs in Sao Paulo and at the University of Arkansas before going to the University of Michigan. She's interested in really big picture questions of flowering plant evolution, focusing on systematics, biogeography, and trait evolution. She has published an impressively long list of very interesting papers in journals, including Journal Biogeography, Evolution, New Phytologists, and Proceedings B. So today, Thais will be talking about plant traits and biogeography. I'm really looking forward to the talk, and I hope you folks are as well. So without further ado, Thais, welcome, and can we give a brief welcome to Thais?
Well, thank you so much for the very nice introduction, Wes, and also for inviting me to give this talk. Oh, I just have to stay close. Oh, I see. Sorry. So my talk today has this title that is kind of a little bit broad and ambitious, discovering the rules of plant biogeography using a trait-based approach. But I hope that by the end of the talk, I can explain this title a little more. And it's basically, I'm interested in ways to understand generalities of how plants diversify in space in relation to their traits, and how can we identify generalities and understand also which are the exceptions, with a particular focus on flowering plants, which is an amazing lineage of plants with over 300,000 species. They are the basis of our diet. They are everywhere in the world. If you look outside now and see a green thing, it's very likely that it's a flowering plant because they are everywhere. So it's something that maybe we don't think super like consciously a lot of the time, but they are basically the organisms that make the biomes as we know them. And basically, even when we are talking about different types of biomes, we are talking about characteristics or clades of flowering plants that dominate that landscape, right? So for example, when we talk about broadleaf rainforest, we are talking about broadleaves, which is a trait, a plant trait, a flowering plant trait that dominate that landscape. When we talk about grasslands, we're also talking about the flowering plant clades that dominate that landscape, the grasses or poaceae. And because we have this intuition, we intuitively know that flowering plants, different traits of flowering plants are particularly more diverse in certain types of environment and climates. We can even tell what the climate of a region is like just by looking at the flowering plants in the landscape. So for example, here we have photos of landscapes in 4 American states. And you can probably guess what the climate is like in these regions just by looking at the flowering plants in these landscapes. You can tell that like Michigan's temperate because it has a lot of this deciduous forest, or like areas like South Dakota is dry and cold because it has a lot of this grasslands, a lot of succulent plants, probably like a drier environment, and this more closed canopy broadleaf forests are probably more tropical or humid. So we have this intuition that an uneven distribution of flowering plant characteristics in different habitats exist and probably result from the evolution of plant traits to better survive and reproduce in in particular types of environments. So some traits are more obvious than others, but what I want to emphasize in this talk is that even like less understood traits probably follow this trend. So how do we quantify trait-environment correlations of this kind? So let's first understand what are the circumstances that may lead a trait to become particularly more common in a given environment over time. So here, for example, I have an environment gradient represented by this polygon. It can be like temperature or elevation. And we have a few species with different traits distributed in this environmental gradient. So you see that species with trait X, which are the little blue circles, are more diverse in this side of the environmental gradient, and species with the trait Y, which are the yellow little circles, are more diverse in that side of the environmental gradient. So this distribution can arise from one sort of migration from one lineage with yellow traits to one side of the environmental gradient, and one lineage with blue traits to the other side of the environmental gradient. And then this unique event where then the whole radiation of blue traits diversify in one side and the whole radiation of yellow traits diversify in the other side. It can also arise from convergence and parallelism, which is like you have many, this is just a schematic representation, but you have many events of migration and trait evolution. And every time that group migrates to a biome, to a different area of that environmental gradient, it evolves that particular trait to better survive there, or maybe it evolved the trait before and then allow that evolution of that trait allowed the movement. But the important thing is that in this case, there to the right is where we observe the strongest support for correlation between trait and environment, because in that case, we have different replicates, like natural replicates of that event of interest. Whereas in this case, you have, this case close to me, you have just one event of migration trait evolution. So basically, trait environment correlation analysis are these ways of discovering general patterns in plant biogeography by quantifying to what extent these unrelated lineages, that's the most important part, have similar evolutionary responses to a given environment. And this is part of a synthesis that I wrote on this topic and published this year on the American Journal of Botany. So in this talk, I'm going to give two examples of recent work of mine that kind of exemplify this concept. One relates to seed dispersal in relation to climate, and the other relates to life history strategies in relation to climate. So I'm going to start with seed dispersal in relation to climate. That's part of a paper that was actually published in 2021, but it took a while to be assigned to an issue. So it's actually like formally 2023, where we explore how different seed dispersal strategies relate to climate in flowering plants. And seed dispersal strategies are interesting to me, especially when we look at fruit type, because they are not only important for plant reproduction, but they are also important for plant movement, because plants are sessile. So they only move, they mainly move during the phase of seed dispersal. So it has an impact on how plants move across environmental gradients. So traditionally in flowering plants, we have types of fruits categorized in two different types. So we have fleshy fruits, which are like drupes, berries, and pomes. And we have dry fruits, follicles, capsules, and achenes, just to give a few examples. This categorization is interesting because it has also some ecological meaning. Because typically, not always, because biology is super complex and there are always exceptions, but typically seeds of fleshy fruits are dispersed by frugivores, such as birds and mammals. So the fleshiness of the fruit evolved as a way to attract these frugivores, and then they disperse the seeds. And typically, on the other hand, dry fruits are typically dispersed by abiotic factors, such as wind, water, and gravity. Again, there are several exceptions to this rule, but it's kind of like a generalization that has been well studied in plant evolution and ecology. And the interesting thing is that fruit type has also been, there has been like some recent studies like this two that I exemplify in this slide that have found that there is a gradient in how, in which type of fruit is most diverse in depending on characteristics of an environment. So for example, in that paper, Chen and collaborators 2016, they found that fleshy fruits are particularly more common when they analyzed distribution of fleshy fruits and dry fruits in Australia. They found that fleshy fruits tend to be more common in lower latitudes, in areas that are warm and more humid. And we found the same thing in that paper, Reginato and collaborators, including myself, for Melastomataceae, where we found that for the neotropics and Melastomataceae, lineages with dry fruits tend to be more common in the open, drier and cooler environments of Southeast Brazil. So we see that there has been studies that focused on this gradient of type of fruit. And there is also theory that kind of explains this potential uneven distribution. So basically, the theory says that fleshy fruits may tend to be more common in warm and humid environments because warm and humid environments tend to promote growth of larger trees. So you have more like closed canopy environments, and then closed canopy environments tend to be more shadier, like shadier environments. And shadier environments require seeds to grow larger, to evolve to be larger, because they need to retain more nutrients. They need to store nutrients for germination. Because the little plant would take, the seedling would take a while to be able to photosynthesize by itself in this shadier environment. But larger seeds, they have a lower dispersal potential on their own. So it's in these scenarios kind of advantageous to be dispersed by animals, because then the animals would tend to take the seeds further away, and then we'll avoid competition with the parent plant. So there is some theory behind that, but then you may ask, but hey, I see fleshy fruits in open areas all the time. So how strong is this pattern anyway? And that's the type of question that I want to answer next. So basically the question that I wanted to answer in this study is like, lineages of flowering plants with different modes of seed dispersal have different climatic niche optima. So they have different distribution depending on their fruit type. And we hypothesized that lineages that depend on frugivores, so the fleshy fruit ones, shift to mesic optima, so higher potential for vegetation growth. In other words, higher temperature and higher precipitation. And lineages that do not depend on frugivores shift to xeric optima, so lower potential for vegetation growth, lower temperature and lower precipitation. So to do that, we selected, first we need multiple natural replicates to understand if there is like a consistent pattern. So we select 5 clades, 5 families of flowering plants for which we had good phylogenetic data available. So Apocynaceae, Rosaceae, Ericaceae, Melastomataceae, and Solanaceae, which combined sum up to almost to 5,564 species. They are also, these 5 families also have cosmopolitan distribution, although with slightly differences. So for example, Melastomataceae there is mainly tropical, whereas Ericaceae is more diverse in temperate areas. And most importantly, all of them have independent origins for fleshy and dry fruits and multiple events of evolution of dry and fleshy fruits inside them. So for example, in Ericaceae, you have lineages like rhododendron, which have dry fruits, or vaccinium, the blueberries that have fleshy fruits. So the next step, we basically downloaded all occurrence points for the species included in these 5 phylogenies from GBIF. And then we corrected, we filtered these points for common problems that GBIF has. So basically outliers in distribution, points on the sea, wrong countries, inaccurate coordinates, etc., using some custom R scripts. And then we overlaid that, and maybe I'll go straight to this slide and go back later, but just to explain more or less the pipeline, we overlaid those filtered points on top of these climatic layers. So for example, here for temperature, And then we extract that value for that climatic layer in that point. And then we calculate the mean for each species based on those values, and also the within species variance that we use as an error term for the analysis later. And then that species means is used as a proxy for the climatic tolerance of that species. So for that part of this work, we used 2 layers that were based on the hypothesis that we had. So we used the aridity index, which is a proxy for humidity. It's like a cool layer because it kind of like, it's like a proxy for potential vegetation growth and also correlates to precipitation. And we used mean annual temperature to test whether they are in warmer or a cooler environment. So we have one discrete trait, which is fruit type, and we have one continuous trait that is a climatic niche. So to combine these 2 types of traits, we used first a discrete trait evolution model to model fruit type in the quorum, or our package quorum. And this is kind of a cool model for discrete trait evolution because it uses hidden Markov models. So instead of having the model basically, instead of scoring all the diversity of fruits in terms of fleshy and dry, it transforms that into a 4 state where you can have variation in transition rates between fleshy and dry in different areas of the phylogeny. So for example, if you have this lower rates of transition between capsules, achenes and berries than you have from capsules to berries, it can potentially capture that, because it allows this uneven rates of transition within the phylogenies. And then we used the quorum framework to map the evolution of the discrete traits. And then we used a continuous trait evolution model in the AUI-R package to model the climatic variables as a continuous trait on top of that discrete trait. So basically, these OU models, they allow us to estimate some parameters of interest, but I'm going to focus on theta, which is the adaptive optimum. And basically, we are testing here whether lineages with different fruit types will have different values of theta in models that allow theta to vary between fruit types. So, so for some results, which is interesting. So these are some of like part of the results of that paper. Don't have time to talk about all of them. So I'm going to talk about mainly the, which I think was the coolest part, which is that we found support for theta to vary between lineages with different fruit types, where lineages with fleshy fruits tend to have warmer climatic optima than lineages with dry fruits that tend to have cooler climatic optima. But what I think is the most interesting thing is that if you look at each of these families individually, you see that actually all the climatic optima, for example, for Ericaceae, which has a more like montane and temperate distribution, are lower than Melastomataceae, so they tend to be in cooler environments. But even within this clade, you have a separation where fleshy fruits tend to be in warmer areas than dry fruits. And that's the same for every clade that we analyzed, no matter where they are in the globe. So for aridity index, we have less strong support for generality because we found the pattern in 4 of the 5 clades. So we found that fleshy fruits tend to shift to more humid environment in 4 of them, but we have an exception in Ericaceae where we have lineages with dry fruits shifting towards humid optima. So one thing that I find interesting about this approach is that it doesn't only allow us to understand generalities, but can also point out to exceptions to the rules for further studies. So for example, in this case with Ericaceae, we think that maybe the trend may be driven by one genus with like a weird distribution in the counterintuitive distribution like Dracophyllum, because it's a genus that has dry fruits and it's distributed in very wet environments. So it's kind of cool because it allows us to formulate hypotheses for future studies as well. So the second example that I have is life history strategies versus climate. That's part of a paper led by James Boyko that has just been accepted for publication in Phytologist. And much like when talk about types of fruits, we also have a lot of variation in how flowering plants, in the strategies of flowering plants to survive environmental challenges. So for example, they can lose their leaves to avoid freezing conditions, or they may evolve succulents against drought, or they may evolve underground systems against fire. So these are all forms of different types of life history strategies to avoid environmental challenges. And some of them have increasingly shortened their life cycle, so that germination, fertilization, and seed release all happen through the favored season of a year. So for example, some other Arabidopsis species complete their entire life cycle in 6 weeks, and then they release the seeds, and the seeds are one of the most resilient structures of the plant. And so they survive the unfavorable season as seeds, basically. So these two strategies have been also broadly categorized into two types of life history strategy. On one side, we have annuals like Arabidopsis, like a lot of the Arabidopsis, that have the full life cycles in a single year, and they die after reproduction. And on the other hand, we have perennials, which include all of the other examples that I mentioned before. They have their full life cycle can last many generations, many years, sorry, one generation, many years. And they usually don't die after reproduction. So as well as with fruit type, there is also a tendency of plants with different life history strategies to be more common in different types of environments. So most of the diversity of flowering plants is of perennials. 90% of the flowering plants are perennials. But when we look at the distribution of them on the globe, we see that some regions have 60% of their floristic diversity of annuals. And these regions are mainly deserts and areas with Mediterranean type of climate. So there might be something there. They are these regions that I highlight with the squares that have higher proportion relatively to the other ones of annual species. And this trend again was noticed before by previous studies. And in fact, you have a few studies, for example, these ones that I'm showing, that analyzed the specific lineages and found that for some lineages, you have a tendency to find annuals that is more common for annuals to evolve in one type of climate than perennials. But when you put them all together, you kind of have mixed results, because some of these papers, because they study just one group or just one region, they find that it's temperature that drives the evolution of annual lifestyle, or it's precipitation that drives the evolution of annual lifestyle. Or maybe it's seasonality, like higher seasonality. It's more important to drive the evolution of annual lifestyle. It could be any of this, right? So with the goal of testing which of these variables we find the biggest number of lineages following that trend, I guess, is that we wanted to ask this question: which climatic variables best correlate with the evolution of annual and perennial life history strategy in flowering plants when we look across many clades? So for this paper, we used 33 clades, again, that we had good phylogenetic information for, and that represent this variation in annual and perennial lifestyle with multiple transitions between them. This scoring of life history was only possible because the Royal Botanical Gardens Kew released data set recently where they have a life history strategy scored for a lot of relatively a lot of flowering plants. So we were able to score many of the tips of these phylogenies and complete with information from the literature where we didn't find them in the WCVP data set. Again, we use the same pipeline. We download GBIF occurrence points for all the tips, all the species in those phylogenies. We filtered them for common problems in the GBIF dataset. And here we analyze 6 climatic variables that have that are linked to our main hypothesis. So we basically want to understand if it's weather, seasonality, or precipitation temperature that drives the evolution of an annual life history strategy. So we analyze temperature seasonality, precipitation seasonality, mean annual precipitation, precipitation of the driest month, and for temperature, mean annual temperature and maximum temperature of the warmest month. And the reason why we include those two is that those extreme precipitation temperature, it's because annual seems to be thriving in desert. So those two variables are a description of like potential for drought with precipitation of the driest month and potential for extreme heat with maximum temperature of the warmest month. We use the same pipeline as I described before to calculate a mean for each species. Again, I forgot to say earlier, but all the codes that we used are available in for this presentation, 2 GitHub repos. Sorry if I didn't mention that before, but I can, if you're interested, just ask me and I can send you the link. And for this, in this case, we also use the combination of discrete trait evolution models and continuous trait evolution models, but very similar pipeline as before. But for this one, we use the new developed model that jointly estimates the parameters of the discrete trait evolution model and the continuous trait evolution model. It's been published by the first author of this paper recently this year, and it's also available in the R package. And basically this framework is interesting because it allows the discrete trait evolution to influence the evolution of the continuous trait and vice versa when we are estimating these parameters. And to summarize the result from all of the 33 clades, because we have so many clades this time, we built a backbone phylogeny for them, for the 33 clades. And we used phylo t-tests to basically test for the significance of whether theta for annuals is different from theta for perennials. Again, theta is the adaptive optima as estimated from the AWI framework. So let's look at the results. This is the result for seasonality. And basically here in the y-axis, we have theta, actually expected mean, but it's a transformation of theta. And what we want to see is many lines of the same color, because blue lines means that theta of annuals are higher than theta of perennials, and red line means that theta of perennials are higher than annuals. So in this case, for example, we have no strong support for general correlation between life history strategy and seasonality. For precipitation variables, we have strong support for correlation between life history strategy and precipitation of the driest month, where we have that in 22 out of the 33 clades, the theta of perennials is higher than, so they have a higher minimum precipitation of the driest month. So they are in areas less prone to extreme drought than annuals. And then interestingly, for temperature is where we find the strong support for correlation between life history strategy and temperature, where in 25 out of the 33 clades, we have that perennials are in lower temperature than annuals. And for 32 out of the 33 clades, which is the biggest generality, we have that annuals are in areas where the maximum temperature of the warmest month is higher than for perennials. So with that result, we kind of explain that, so okay, so it's maximum temperature of the warmest month that is probably driving the evolution of annuals in most clades, right? So we hypothesized then, it's something that I hadn't mentioned before, but probably most of the annuals in our data sets may be winter annuals, which are the annuals that germinate and reproduce during the winter, because we found that probably they are in this in areas prone to extreme heat because maybe annuals can evade the hottest season in the form of seeds, which is one of the most resilient structures, plant structures. So it's possible that in both annuals and perennials, the adult, like the mature form, is equally sensitive to heat stress. But then because annuals can basically die before the warmest season, they can just release the seeds and that seed survive for the next year. So the take-home message of my talk is that particularities of each lineage matter when studying trait-environment correlations. And of course, that's why we see that not all clades follow the same trend. But I argue that I think this makes all the more remarkable that also some profound generalities exist. So for example, for seed dispersal, all 5 analyzed clades presented the same trend for mean annual temperature. And for life history strategy, 32 out of the 33 analyzed clades presented the same trend for maximum temperature of the warmest month. So just to finish this talk, I just wanted to say personally what I think is the next 4 trait-based approaches to plant biogeography. Most of this, of what I presented in this talk was mainly data analysis. It was what I was doing in the last 3 years, but I'm originally a tropical botanist. So kind of excited to be able now to go back to the field and collect more data. And I think that's very important for the approaches that I presented here today, because as you saw, having good phylogenies and good trait data sets and good data sets of occurrence points is fundamental to answer this type of questions. And unfortunately, for for phylogenetic representation and a lot of traits that we use that are important for the ecology of flowering plants, like woodiness or seed size, we have very limited information from tropical areas. This is an example, again, from the synthesis that I wrote this year, just like plotting distribution in terms of how much you can find from common open data sets of traits in relation to species richness in each of those regions. And we see a strong gap in data from tropical areas. So it's basically, this means that we are trying to understand, because like many other groups, flowering plants are most diverse in the tropics, and not having good data from the tropics means that we are trying to understand this puzzle with the main pieces missing. So yeah, I want to thank my co-authors. If you have any questions, feel free to send me an email. Thank you for the funding, and yeah, thank you for listening. Thank you.
I think we have some time for questions. So Matt and I will have microphones. So just raise your hand. We'll come and find you. And if there's any questions on the Zoom link, we can answer those too. Thank you, Thais.
It wasn't clear from, so at the beginning you mentioned the importance of phylogenetic independence of, like contrasts between traits and climates and so on, but it wasn't clear. So like for the analyses you did for a single clade, mm-hmm. was that quantifying the correlation or not?
Right. Yeah, so for each individual clade, we also have independent origins within each of those clades for the traits that we were interested in. So in both cases, we basically analyzed the correlation within each of those clades, and then we compared the direction of the correlation among all the clades to see if they all kind of tend to have the same. In this case, we're using the AUI framework, so the OU model, so just see if they have different, the theta parameter shifting towards the same direction in terms of the discrete trait of the fruit type or life history strategy.
So do you think your results would have been different if you simply just ignored phylogeny and you just like use the geographic data, the climate data, and the trait data? Like it wasn't clear to, like the way you presented it, it was like, well, we got, theta, theta dry versus theta fleshy, and compared them across clades, but it wasn't clear like, what I guess I was just wondering like what information was the phylogeny giving you? You know what I mean? Because it seems like you're comparing. It seems like you could have just estimated, climate values and trait values just from geography or something. You know what I mean?
Yeah. Well, in the first case, that's a good question. I don't know because I just use the phylogeny, but yeah, that's true. You mean like if we just get the mean and then compare?
Yeah, like it just wasn't clear to me like what information the phylogeny was providing, because you didn't present any like regressions, phylogenetic regressions or anything. So I'm just wondering.
Oh, I see. Well, the OU models, just like a model of trait evolution that we fit into the phylogeny and the information from the tips, and then estimate based on like estimate parameters of the optima, adaptive optima for the different discrete traits to have like the discrete trait mapped there, and also variance, and also has another parameter, which is the pull to the optima, has 3 parameters, and we basically just estimate these parameters that maximize the likelihood of observing that distribution in the phylogeny.
Right, but those parameters they don't figure into your results, right? I mean, you're just presenting like, here's theta dry versus theta fleshy. And I guess I was just wondering, like, if you just measured like, the species distribution of a dry versus fleshy, you'd get different, you know what I mean? You get different environmental values, you know what I mean? It's like, it just wasn't clear to me, like, why you need the model, why you need the phylogenetic model, right? Because you could just kind of estimate from extant distributions what the values are.
Yeah. Well, one example would be like if you just get all the data for like climatic data for every species and then trait data for every species, and put together and run like an ANOVA or something like that. You may not find a strong correlation in that case, because if you don't use the phylogeny, you're basically assuming that adaptation is instantaneous when something moves towards that different climate. You don't have like the relationships between them being an important component of the analysis. So you may not find a strong correlation simply because simply because if you don't use the phylogeny, you're assuming that you're assuming that once a species changed to that environment, then trait evolution follows immediately. We're not even talking about trait evolution, it's just like trait change happens immediately. But if you incorporate the phylogeny, you consider this kind of lag that comes from the phylogeny as well. So I would guess that would be one of the importance of the phylogeny.
Yeah, I guess another perspective you could take is like, instead of considering like phylogenetic replication, you consider like geographic replication, like, so the succulent biome on different continents, you know what I mean? As kind of replicated cases of assembly of floras with different trait, you know what I mean?
Like, that would be ideal if I could also have a way to quantify if I'm getting this data from different environments that are not like disjunct biomes that are kind of similar, right, in the world. That's also a good point, but I'm not sure how to do that yet with this approach. So yeah, thank you.
Hi, yeah. I was thinking about a question about how you're defining seasonality, because there's actually, if you're looking at a global from a global perspective, you've got two different kinds of seasonality. You've got the, the temperate seasonalities where you've got summer and winter and the temperatures change radically, but also if you're in tropical and subtropical regions, a lot of times what you're looking at is a wet and dry, well-defined wet and dry seasons, which is a different type of seasonality than the temperature connection. So I was wondering, did you ever look at anything that compared the two different types? So if you're looking at groups within, say, the tropical region that have, the certain groups where certain parts of the family are in the wet zones and certain parts are in ones with well-defined dry seasons where they're going to have to, would fight that. Did you ever look at a comparison between that and the temperate, non-temperate comparison? I mean,
yeah,
you should be able to find families where you can do that.
So you mean for the second study, we used also precipitation seasonality as one of the variables. So I guess, I don't know if that would, is that what you mean?
Well, yeah, because I was wondering when I was looking at it, whether temperature and seasonality were actually that independent as variables. And then I started thinking about it in terms of this tropical and non-tropical regions, but then you could compare the two types if you wanted to look at your data, if you have the data for that, which I would think you do. You could look at the two different types between a seasonal, a temperature-related seasonality and a precipitation-related seasonality.
Yeah, we look, you mean this too?
Yeah, but I don't see how they're not. Oh, okay. Let's see. That one's there. All right, okay. Thank you.
So there's a question. So one of the things when I'm out there, like in the field, one of the things that I notice about those traits is that they influence like the abundance, right, of the species that we see. So it's not only that I have more species with with fleshy fruits necessarily, but they might be more abundant as well. Could you tell something about how abundance is related to diversity in those trait differences in plants? Because usually, in my impression is that we define the biomes by what's common, not necessarily how many species have a certain trait or not, right? So I don't know if you could elaborate a little bit more on this.
Yeah, that's a very good question. We definitely, define the biomes by which traits are more common, maybe more abundant. It doesn't matter how many species you have there. In the case of the work that I presented, most it's all about species richness, though. It's all about diversity. I didn't consider abundance as one of the variable of that trait, because I don't think we have a good way of measure that in this scale. I may be wrong, but and I don't know how I would incorporate that in the analysis either. But it's true that when I talk about how diverse a trait is in one region, I'm talking about in how many species that particular trait appears in that region. It may be that all of those species are rare, but and then it's still you have that region like being highlighted in the map, even though it might be like all rare species. So yeah, that's a good point.
That's the case for any of the traits you work, or is diversity correlated with abundance?
I don't know. That's a good question. I think in some cases probably is, and others is not. Yeah, I think there is a variation too. Yeah. Yeah.
Thank you, Thais. That was a wonderful talk.
Thank you.
I wanted to ask you to talk a little bit about any links that you see between this kind of research and your work as curator of the herbarium.
Wow,
Are they,
that's a very good question.
how related do you see them as, how different? So for example, in this talk, you mentioned lack of trait data as a big problem. Is that something that your work with the herbarium or herbarium work in general can help resolve? I wonder if you could just talk about this.
Super, that's a great question. Yeah, because a lot of this data, DNA data, trait data, occurrence data, all come from DNA data. Often we go to the field to collect fresh specimens for that, but trait data and occurrence data all come from herbarium specimens. So yeah, so basically it's the basis for a lot of the research that I do. I think herbarium specimens are still underutilized sources of this type of trait data. And I've been talking to a lot of, in my meetings with folks today and yesterday about this, about how also I wish that there were better ways of standardized trait data collection from herbarium specimens, because sometimes I feel that in these open trait data sets, yeah, I don't know, that kind of combined data sets from different sources, people measured these traits in different ways, and it would be nice to have like a protocol to standardize that. Also, occurrence data all come from herbarium specimens, so also I wish that there was a way to, after cleaning and curating occurrence points, we could return this information somehow to GBIF, for example, because then with time, GBIF would be probably improved over time if there was like an easy way to just return that to GBIF. So, but yeah, herbarium specimens are the basis for all the data that I use. So, and we'll continue to use as a, and I don't know, I intend to also improve, like provide new specimens also to the collections in Michigan, because we have an amazing collection there, but it needs, I think for some groups that I'm interested in, it needs more recent specimens. I think there are a lot of very important collections, but they are also quite old. So for like, if I want to do, I don't know, destructive sampling of any kind, it would be nice to have like new specimens. Also for like, I don't know, herbarium specimens may be used in ways that we can't even imagine in 100 years from now. So I think it's important as part of my job to make sure that we have like, keep collecting new specimens, and also like making sure that we have these snapshots in time from, yeah, at least the groups that I'm mostly focused on. Yeah. Yeah, so many ways, yeah.
Just want to say thanks. That was a really wonderful talk. I had a question I was hoping you might be able to address. I'm wondering, in both these studies you highlighted, to what extent do you see the transitions to the more derived state? Are those transitions somewhat clustered or concentrated in time, or do you see them somewhat evenly distributed through time? And then are you able to relate them at all to what's going on in like the paleoclimate, geography, or ecology at all?
That's a good question. I don't remember for the second, for this study on life history strategies. For fruit type, well, I think the main thing that we discussed about that in that paper was related to like evolution diversification of frugivores, I think. But most of the clades that we analyzed were kind of more recent, I think, than the estimates for the origin of like the first fleshy fruits and like fleshy fruits frugivore interactions. Yeah, trying to remember now. what was like, if there was like anything specifically. Like, I don't know, I kind of think that within each of those clades, there was no like clear trend of where most transitions happen. So yeah, yeah, really.
Thank you.
Yeah, thank you.
I just have one quick question, and then I guess we can probably wrap up. Thais, very nice talk. I have a question that is related a little bit to what Matt was just touching on. It has to do with transition rates, which I know you estimated as part of this. In fruit type, for example, also with life cycle, but for fruit type, for example, is there a directionality bias in these transitions, kind of apart from habitat type or anything like that? Certainly in some situations, we have a non-reversibility of transitions in certain certain pathways. And I was wondering if that might be the case, for example, in Ericaceae, if you transition to, let's say, dry fruit in that family, is it really hard to go back to having fleshy fruit or vice versa? Or if you have a general pattern of that across the groups that you looked at, and if that could bias any of your interpretations, maybe.
I see. Yeah, I think for most of the groups, dry fruits is the root state, and you have a trend to evolve dry fruit, fleshy fruits from dry fruits, so like dry fruits to fleshy fruits. Except I think Apocynaceae, the root is fleshy fruit. And yeah, I think if like these ancestral state reconstructions are not very good usually. Like, I don't know if this is contentious. It's like, it depends on like how broad is your sample of species as well, can change what happens towards the nodes that are closer to the root. For example, if we include the sister group or not, sometimes it changes what is going on in like those older nodes. So I always find it a bit difficult to talk about like, I don't know, it's so important though for all this modeling framework, the transition rates, but yeah, kind of like walking in circles. It was just to say, maybe there is a trend from dry to fleshy fruits in most clades, except Apocynaceae maybe, which is fleshy to dry, apparently.
Great, thanks. Okay. All right, I think we are good. Let's give a round of applause again to Thais for that talk. And thank you all for coming.